Hey, Matt, are you there? Oh, Ken's there. There's Ken.
Hey, people are showing up.
Hi.
That's great.
Oh, yeah. Hey,
Good.
was that you, Trevor?
Yes, indeed. I just wanted to touch base because I emerged from teaching for a few minutes and I thought I would pretend I'm still a researcher.
Awesome. What's up?
Not much. I just wanted to say I didn't sign up for CCFR this year because of teaching, but
okay.
I'm just like
I can send you
completely
the talk if
swamped.
you like. It's just an MP4.
Oh no, I'm just saying thanks for pulling that together. That's all. It's great.
Yeah.
And that and the postdoc and I, Andy, are going to polish off that thing during reading week next week to send you guys the draft of the intro. It just sat for a while because of teaching. I'm prepping my first year biology for the first time this year, so I'm falling behind on that.
Oh wow.
Yeah, I've once again, it's going to be great next year. Just this year is a bit of a gong show because it's a full prep. So, anyways, I'll send it to you next week with Andy. Okay.
Sounds great, Trevor. Thanks.
All right. And if you send a copy of the talk, great. I'll definitely watch it. But I just want to say thanks for doing that.
Okay, no problem. It was
Right.
fun.
Well, catch up later.
Hey Nick, how you doing?
Oh, I'm great, thanks. How are you, Ken?
Oh, good. Reading week, you couldn't have come here fast enough. I'm looking forward to it.
Yeah.
Not that it's going to be any easier, it's just not lecturing for a week.
I guess we consider ourselves lucky that Doug Ford did not move our reading week with the March break.
I forgot about that.
Well, I've got a meeting at 4 o'clock with my colleagues Laurentian because I'm adjunct there, and I feel bad for those people. They're in a tough bind right now.
Yes.
That's awful.
Yeah, I gave a seminar to that last week, and they filled me in on the details, and it's quite gory, to be honest with you. So all of our create money was spent by the operations budget. So it was, literally our create grant's gone. They spent it on operations, which is hard to believe. Yeah. Anyway, another story for another day.
Glad you could make it, Vince.
Yeah, happy to have been a fly on the wall there. This is a really very, very cool project. We're hoping to access some of this expertise for some of our studies, as you know. So this is very cool.
Oh, fantastic! Yeah, we want to get to the field testing and the field validation, but to do that, we need to finish activity 3 first, and so that's going to be the real all of my effort really for the next 6 months.
Yeah, I bet.
Trying to get something there, and so yeah, it's exciting, but yeah, it's a daunting amount of work still left in.
Yeah, and here we are trying to push you into other things like doing it in mucus and other things. So push it harder.
I'm a big believer in if we can develop these types of assays for non-lethal testing, then that'll be the best way moving forward.
Absolutely, I agree.
So, Ken, was there a specific agenda for the breakout rooms?
As far as I know, we're supposed to just organically talk about the activity 3 and activity 4.
Exactly. It's just an open Q&A session.
Ken, I guess I have a question for you. I know in the past we had briefly talked about some of the pros and cons of using blood for non-lethal sampling. And I know you've had some experience with that on previous projects. And it was brought back up again today and made me think that this is perhaps something that we should do in some of the projects that we're going to start this summer. But I wanted to hear from you about some of the limitations that you've encountered.
I haven't personally done a lot of extraction of RNA from blood. I just know that it can be a little bit tricky sometimes. I think it, as a default, just to avoid some of the hazardous chemicals, I try to use a lot of column-based extraction kits in my lab. So I would suspect that it would probably work a lot better if it was a phenol-based extraction at the very least. So from that perspective, I mean, if they can get enough of the red blood cells, it does seem like a really fantastic non-lethal tissue to exploit. I guess you're limited to just what types of responses you're going to be able to detect, but I mean it.
Yeah, but that's certainly the case also for the mussels and the gills anyway.
Yeah.
And it may be that if you have an approach where you take all three of these tissues via non-lethal sampling, that the profile between the three tissues might give you quite a bit of insight. I don't know. It's something I'm tempted to try on experiments that will start this summer. We always collect the blood anyway to measure other things. We just typically don't keep the red blood cells, but if we could come up with a protocol where we could readily get good quality RNA, I think this is perhaps something that we should do.
Absolutely.
We did get RNA out of blood cells. We just don't know what the heck it is. There's a lot of 16S and 18S, etc. But we're planning on sending it off for RNA-seq just to see what the heck it is. It looks like Eddie has a question. Eddie?
I do. Thanks, Dan. So a couple questions. What is planned for assessing the sublethal sampling of fish? I saw that come up. I'd just like a little more information on that to start out with, if we could.
I guess, Nick, you're actually designing the experiments to do this. I base a lot of my assessment of sublethal sampling, because we've done it on a lot of larger fish species, and when we're combining it with telemetry tagging. And so we know, at least from survival post-release, that some of the smaller tissue samples are not influencing survival.
Also, Steve Cook has applied for money to do it at a large scale on a number of Great Lakes fishes, and basically just do the non-lethal sampling and then hold the fish and use telemetry, same as what you're describing, Ken.
Nick, from an experimental perspective?
Well, we just finished an experiment at Alma where we did repeated gill and mussel biopsies from pit tag fish on a 3-week interval over a period of 3 months. We still have to do all of the expression profiling work, but at this point, all we have is just general grow feed conversion efficiency, just typical aquaculture parameters. But we are just starting now to look at the expression profiling. So the idea there was just to see whether we could use repeat non-sampling, non-lethal sampling to essentially look at expression profiles through time within the same individuals. We did essentially, the experiments that we carried out had aquaculture level of densities in the tank, and we would kind of sample half of the fish repeatedly through time. The other half were not sampled, so we had a true comparison within the tank. Like I said, repeat sampling over a chronic period of time in the hope that if we can assess kind of immune genes, cellular stress genes, and other functional groups of genes, that we can get a better sense of whether these non-sampling tools can be used repeatedly on the same individuals, in a aquaculture context, but perhaps also in other contexts.
Mhm.
Okay,
Oliver, do you have a question?
thank you.
Yeah, Dan, I had a really quick question. Just kind of building off your blood transcriptome, you kind of already answered this question, but as sort of a non-expert, do you have any idea, can you give us an idea of what we might expect in the blood? Or do we really have no idea? We just have to screen and then see what we're going to get?
Essentially, I've got no idea.
Okay, no problem.
Brian Dixon and I have been chatting in the side about white blood cells versus red blood cells. The idea is that it might be the white blood cells are functional, but the red blood cells aren't. The fact that we were able to get some we've got some RNA out of just a mixture. It was just the packed cells, so it was white and red blood cells. You just don't know. And a lot of the fish genomics, it was never actually tested, but everybody knows that such and such a thing happens or doesn't happen. And then when we try it, we find, okay, wait a minute, something different. So I'll be curious to see what happens, but I'm not overly optimistic we're going to get a wide range of genes that are being expressed in the red blood cells.
No worries. I mean, I was just curious, and you kind of already answered that question for me. So just another quick question on the sublethal sampling. Again, I'm a new person to fish, so is there such thing as a sublethal sample on liver tissue where we can open the fish up if we're putting a transmitter inside, take a biopsy? I think I have just realized the answer to that. Likely no.
I'm laughing. I've brought this up with a colleague of mine, and I'm convinced it's doable. I'm sure, like when we're doing a telemetry surgery, we could just take a little tiny piece of liver, and I bet you it'd work. And I'm trying to convince them that we should test this out in our basement facility.
Okay, I mean, it's definitely something, as someone who's interested in energetics and metabolism, it would definitely be a great thing, sort of, again, as a non-expert. I don't want to go in there and sacrifice a bunch of fish necessarily, but if the team can suggest, here's sort of the minimal size of fish, here's sort of the minimal size of sample that we would need to actually get enough material out of there to do what we need to do. And then building off of that, I'm really interested to see, I understand that we would expect different genes to be turned on, for instance, in the gills versus liver versus muscle. But is there any expectation whatsoever of being able to do a muscle or a gill biopsy and have it correlate with something back to the liver, so you don't actually then have to go and do the liver biopsy? Or is that just basically dreaming way too much?
I think that's a great approach, and one of the ways during the early validations, if we can get those multiple tissues and try to figure out if there is an expression profile in a non-lethal tissue that does match a general pattern in a lethally sampled tissue, I think that would be a great way to move forward with validating some of these approaches. Just to follow up on one thing though, with the red blood cells, there was a microarray study a while ago on thermal stress in red blood cells, and so at the very least it seems to respond quite well with a heat shock response. So they were able to detect a lot of heat shock protein upregulation based off of the thermal stress in one of the salmonids, probably rainbows. Anyway, sorry, to get back to your other comment about whether we could figure out if a Gill transcriptome can represent something that's going on at the liver. Does anyone else have a thought one way or another? Nick?
Yeah, so the experiment we just completed on this non-lethal sampling, we had parallel tanks that were exposed to exactly the same treatment where all the fish were sacrificed, and we took the livers. And we also took the gills and the muscles from all these animals that were sacrificed in order to do exactly that, do side-by-side comparisons in animals that were euthanized as opposed to animals that were repeatedly sampled for non-lethally. And the red blood cells work on heat shock proteins done 15, 20 years ago out of Queen's University, Bruce Tufts' lab. That was all done on red blood cell, and that was all heat shock protein. So yes, the red blood cells have been used extensively for heat shock protein work, but to my knowledge, not much else.
Okay, great. Thank you so much. And I completely understand. I don't expect the genes in the liver, for instance, to be the same as in Gill's. But I mean, if there was a correlation that was consistent, then I could just use one as a marker and expect the other one to be increasing. So that's good. I'm really glad that everyone's looking at that. Thanks very much, everyone.
No worries.
The other thing that Brian Dixon brought up is a syringe biopsy. The nice thing about using the open array or the nanofluidic approach is you don't need a lot. So if we sort of shift our view from the old, type of RNA extraction where you need a huge chunk of tissue and go into micro extractions, then we might be able to get away with a needle biopsy.
Yeah, that's a really cool idea.
Of the liver.
Yeah. Other things maybe not so easily. Eddie, your hand is up. Do you have a question?
Sure. Thanks again, Dan. So actually, as a follow-up to what you just said, I was going to inquire, particularly around muscle and gill samples, what sort of minimum sample size you're thinking, tissue weight, I guess, you're thinking for both of those two tissues in particular?
Ken?
For gill tissue, you don't really need much, especially depending on the size of the fish. You have a little bit of variability of how much you can take. Usually we just go for, about 2 to 3 millimeters off the tip of the filaments. And so it's not a lot, and at the very least, and there's properties of the gill that make it really, really good for extracting enough RNA with tiny samples. It's a little bit harder with muscle just because the nuclei aren't as densely packed.
Yeah, I would say 50 milligrams is about all you need from either tissue.
15, was that?
50.
50. Yeah, thank you.
Celine has a question about how do we expect to validate the chip once we have it up and running.
Yes, exactly. I mean, we would need a way that it could be really, useful for you, and just having an idea of how you see that. Or if it's too early to think about it, I don't know.
Celine, stop asking hard questions. I think what you have there is great. If we can sort of have the already established methods within the lab to verify that they're responding to an experimental treatment, and then to compare that with the results that we get off of the STP chip, that would be off the top of my head, I think that might be a way to do it, especially for the experimental work in activity 4. But I guess this is more of a Daniel question as well.
Yes, Celine, the validation is going to be critical, and it's going to be dynamic, because I think that we could do a number of mesocosm studies or lab studies, and then the PIs involved in activity 4 would say, okay, yeah, this is working nicely. We hit them with temperature stress or immune challenge, and things are doing what we expect them to. I think there was a question earlier on that led to, fed into your question, and that is, what point will you be able to actually convince regulators that this is reliable enough to use it as a gold standard? You go in and test a fish in an environment and come back and say, okay, there's no evidence of any stress, so the mine isn't affecting the water system. That is where the partnerships with everybody from Environment Canada to consulting companies is going to be critical, because those are going to be the types of validations that are perhaps going to be accepted. Oliver, you had your hand up for a second.
Yeah, and I don't mean to be hogging things. If someone else has got a question, please jump in. But I just had a quick question again about blood. So if we're interested in, maybe this is something that Brian Dixon can jump in and sort of answer. If we're interested in, the immune component of that whole blood. So, as a crude, ecophysiologist, when I spin down our blood, we get what I call crudely as our Buffy layer in between. But, is there a reason, if we're really interested in the immune system, to try to isolate that away from the whole blood, so that all of those red blood cells and all the other proteins that are in there sort of don't swamp out a potential signal? And maybe a bigger, better question is, do we even expect that immune component to be, transcriptomically active, for instance? Or is this, again, I don't know, so I'm asking a sort of a pie in the sky kind of question to see whether this is worth us going after.
In the lab, I use Buffy coats all the time. So yes, more or less. Yeah, I do try to screen out the red blood cells, because again, the question of whether they're transcriptionally active or not because they're nucleated is up in the air. But quite often, I just want to get rid of all that excess protein and junk that's going to mess up what I'm trying to purify.
Okay, so maybe, that, again, for us non-experts that do have access to systems, we won't want to mess this up. Maybe if there's a simple, quick protocol we can basically use to separate out those cells, just so we don't end up with junk at the end of the day. And then, of course, we could have done a simple step. That would be really great to have.
I can get it. You have to use a slight different density of Percol because the red blood cells are different than the non-nucleated ones. But I have that in the lab. I can circulate that around.
That would be great. Thank you so much. That's awesome. Thank you.
One of the things, Ken and Nick, that strikes me is maybe we should have a separate meeting to talk about non-lethal assays and come up with a strategy, because like the red blood cell RNA-seq that I described, it's just one of my students doing it out of curiosity. Vince, I know your hand was up.
Yeah, I just had a couple comments. First of all, I'm really interested in this non-lethal tool development. I'm also interested for a different reason in non-invasive tool development. So you talked about liver biopsies, etc. And I guess it gets back to my other question about regulatory buy-in for some of these tools. I think we can all recognize the statistical robustness of assessing the health of a single fish over time. That's really much more powerful than subsampling the population. And when you start to do things like liver biopsies, I think you compromise that ability. But if you can come up with a truly non-invasive method as well, you can not only validate exposure, but you can validate recovery of the system as well. So again, I use my example of an oil spill, right? You go to a location, you sample the fish populations there, you can validate that your marker is sensitive as an indicator of exposure, but then you can also use that marker for recovery. And it's a non-invasive way to do environmental monitoring that would be very powerful to regulatory agencies like the Canadian Energy Regulator or environmental effects monitoring for mining communities, that sort of thing. That's just my comment.
That's a really good point, Vincent. And like Oliver's, you moved fish into closed small systems and then extracted cortisol from the water. Oliver, do you want to comment on that? I wonder if we could do messenger RNA the same way. So it's still interrupting the fish, but not invasive to the fish.
Yeah, it's been really useful for us for really small fish, especially when we want to do sequential sampling of the same fish over time. It's been great because it's highly non-invasive. It doesn't take an awful lot of effort. The effort comes in the lab trying to get at the hormone in such a large volume of water. We typically put the fish in, 250 ml beakers. We're working with small fish, of course. You just throw a sturgeon in a big tank. I'm not so entirely sure what you'd have to do there. But yeah, again, I ask these questions because I am not an expert in these techniques, so it's easy for me to sort of ask questions and not be limited by sort of any knowledge of what could or couldn't work. But I think it would be great to give it a try. Why not, right?
Well, it's a very selfish question on my part. Where I work for the Experimental Lakes area. We have these fish populations, which, geez, they almost have names. We have tagged almost all of these fish. So repeatedly sampling fish is something that we can do at our facility. And if you start to conceive of some of these studies that this group is interested in, I mean, think of us as a platform to be able to do those kinds of studies. We have the resident fish biologists who are familiar with those fish populations, can provide some on the ground, feet on the ground to get some of the sampling done. And we're familiar with sampling some of all of the sort of sample types that you are used to collecting. So it's just something to keep in mind as you go forward to the validation step in this process.
And I think it comes back to something you said, Daniel. I mean, having a meeting about non-lethal sampling would be great, but also making sure that whatever techniques we think we should give a try, having a good sense of the collection techniques so that we don't end up with a null and negative. And it's maybe due to, my poor sampling, because again, I'm not an expert. I don't appreciate what needs to be done. If we're going to get no, this doesn't quite work, we really want to make sure that that door is closed, but closed, effectively. And we know that we've got evidence to say that's not going to work.
Yeah.
I think standardized protocol development is a key part of GenFish, from activity 1 to activity 5, so that whether you're going out to non-lethally sample a liver with using a needle biopsy, or you're going out to determine how a particular end user group is going to respond to transcriptionally profiled fish release, that's going to be critical. And there is really a central effort by ARFA and Joe to keep us on track of coming up with standardized protocols and then communicating them. But we do tend to sort of stay within our activities. So we develop an activity for sampling and storing messenger RNA tissues, and that sort of sticks in activity 3, and then we've got to cross the boundaries a bit more.
Just another quick question from me. I mean, is this something in terms of non-lethal sampling that would be a worthwhile small paper to write up in terms of saying, as another way to basically say what GenFish is doing, here's the potential ways that we think we could. And it's kind of a way to say some of these are already tested, but we want to validate them fully. Some of these are untested, but here's the potential to kind of get even more interest in some of these things. Or is that just not worth doing?
If I can jump in here, we have a manuscript in review right now, which is the applications of non-lethal sampling for transcriptomics in fishes, and so hopefully that'll serve as a document. It says this is what's been done, this works, and then this is what potentially could be done and could work. So hopefully that would be kind of what you're referring to here.
Yeah, totally, Ken. Sorry, Ben. Sorry, Ken, I didn't realize that you had that on the go. That's fantastic. Again, I'm always thinking of sort of me as, yes, I'm a scientist, but I'm a non-expert in the field. So any kind of directional review or manual that basically says go this direction for these reasons is always fantastic for someone like me. Sorry, Vince, to interrupt you.
And yeah.
Oh, no, sorry to jump in. There's always a lag. So Ken, you mentioned before about, the prospect of doing liver biopsies, which of course would be really powerful. You can do metabolomics as well as a number of other diagnostics there. And you mentioned about testing it in the basement, but what about testing it on a wild fish population? Because in a laboratory setting where they have optimized, feed regimes, etc., you may not see that competitive disadvantage that results from the stress that's induced by recovery from that surgery. We just had the fish crew at the experimental lakes area just had a paper published on survival and recovery and general fish health after a telemetry surgery, and I think this is the sort of next natural extension on that kind of thing. Is can you do a liver biopsy, and then following that cohort over years, what is their competitive advantage or disadvantage or lack of an effect?
That's a great idea. That would be fantastic, because it's one thing to keep something alive in a lab, as with cushy and they get everything brought to them, whereas what does it actually mean over a course of years in the wild? That would be fascinating.
Mm-hmm. Yeah, the biggest hurdle might be the AUP, but I think you can get past that.
Don't we leave that for you guys to get?
Yeah, actually you do. That could be done, yeah.
But I'm beginning to think that perhaps the non-lethal sampling folks should get together and do a targeted study where we do RNA-seq on tissues taken in multiple ways.
Yeah.
Just to see what can you get from different tissues and whether the non-lethal biopsy biases what you end up getting. Like the process of trying to find the liver with a needle biopsy, for example, as opposed to just killing them and opening them up and getting it in RNA later.
Yeah.
Well, we did a bunch of work there with ultrasound and looking at liver imaging with ultrasound, and that can be done. So we have a poster that was presented at an environmental toxicology and chemistry meeting, and you can get actually a pretty good measure of liver size using ultrasound, just handheld portable units now. So to marry those two techniques would be pretty powerful, I think.
Yeah, if you could guide the needle with ultrasound, now you're talking like amniocentesis on fish. Then maybe you could actually do a head kidney sample. I don't know.
Depending on the time of year, if you're looking at lake trout, if you're trying to do that in the fall, the gonad occludes anything that you're able to see on any of the internal organs, but early on in the season you can do it.
Nick?
I think to me, one of the big unknowns with the non-lethal sampling is just coming up with some kind of, table where you identify how specific tissues are useful for specific types of stressors. Because the gills might be useful for, osmoregulatory challenge, ammonia load, a few different things, but totally useless for other things. And the same is true for the muscle. And they may have intermediate applications for specific type of environmental stressors. But that knowledge, I don't think that knowledge is out there. Exactly how would you use what type of non-lethal sampling, depending on what you're interested in, what your application is. And to me, that's not really clear cut.
I was laughing as you were saying that because I tried to generate a table for that paper and then forgot to upload it when I submitted the manuscript. So it's made, it's just not in review right now. So hopefully a reviewer comments on that, and that'll be an easy fix. But that was one of the goals. I completely agree with what you're saying, and that was one of the goals with that figure that Sonia Mikulewski put together. Vince and I co-supervise her. Is where we have the different types of samples, where they're coming from off of the fish, and what they can theoretically, what types of responses they can tell us if we're interested in those types of processes.
But there's still a lot of holes in our knowledge there.
Yes.
There's in chat and in other ways, I think there's a lot of interest in having a specific discussion. And what strikes me is if we had the full STP chip, that would be perfect. We take tissues under different conditions in different ways and standardize with the STP chip, because the level of variation that you get when you're using the nanofluidic is tiny. So we've got a really standardized way of doing the transcriptional profiling. And Nick, one of the experiences I've had is with goofy grad students who tried the wrong gene in the tissue. We actually detect messenger RNA in the strangest places. Really low expression, but genes that aren't supposed to be expressed in the liver sometimes do show up there. And you got to wonder whether it's maybe the blood cells or who knows. But I think we're at the end of this chat session, so we can continue if we want, but if we won't, we can shut down and everybody get a short break, and then we reconvene at, well, in 20 minutes to do the informal and meet the students and see what they're up to. I'm looking forward to that. Any pressing questions? I agree, Vince. This was really interesting. I had a good time.
Yeah.
Think about if you're putting together a review like that, think about a writing retreat type scenario where everybody brings their ideas and gets it all written down in a Pelston type environment, and you hammer something out on a pretty quick timeline. That would be very useful, I think.
Yeah, great idea.
Yep. Thanks, folks. We'll see you in a bit.
Okay. Bye, all.
All right, everyone, you guys all heading back?
Yeah,
Excellent.
just reading the chat.
Okay, I will stop recording. Thank you.
Thanks, Joe.